Dear students, this is 12th Zoology, Chapter 4, Principles of Inheritance and Variations. Higher Secondary, second year, Zoology. Today, we are going to start new chapter, Chapter 4, Principles of Inheritance and Variations. Chapter 4, Principles of Inheritance and Variations. The insects what you are seeing is fruit fly. That is we call as a Drosophila. So this Drosophila are ideal for the study of genetic and development. So these fruits fly are very useful to study genetics and developments. What is genetics? What is genetics? Genetic is a branch of biology that deals with study of heredity and variations. That is we call as a genetics. So it describes, which means the genetics describes how characteristics and features pass from one parents to their offspring in each successive generations. So what they say here, the genetics, they explain the characteristics and structure which pass from one parents to their offspring, one parents to their young ones in each coming generations. So this genetics only describes what are the characters the parents they have. That characters just they transfer to the next generations. That next generation only they say here offspring, a newborn baby. The unit of heredity is known as gene. The unit of heredity is known as gene. Underline is one word they'll ask you. So here a gene is the inherited factors. Gene is the inherited factors. What is inherited factors? The gene or the responsible to carry information from one generation to another generations. That is, they say gene is the inherited factors that determines the biological characters of an organisms. Variations is the degree by which progeny differs from their parents. What do you mean by variations? Variation means change. The variations is one change. That is the degree by which the progeny differ from their parents. What is progeny? Young ones. So young ones they slightly differ from their parents with help of variations, with help of changes. So in this chapter, we are going to learn about what is multiple alleles with reference to the human blood groups. What is in this chapter we are going to learn what is multiple alleles with reference to the human blood groups, sex determinations in man, insects and birds, sex linked inherited traits, genetic disorders and extra chromosomal inheritance. So these are the topic we are going to discuss in this chapter. The betterment of human race can be achieved by methods like eugenics, euthenics and eugenics. Okay, here the next topic. Next topic is the one multiple alleles. Next topic is the one multiple alleles. Before we enter into new topic, we'll learn what is eugenics and what is eugenics, what is euthenics, what is eugenics, what is euthenics, and what is eugenics. So these three words deals with race improvement through heredity deals with race improvement through the environment. We'll see the meaning of it. Eugenics is a hygiene for future generations. Just if you want to write, you can write it in your textbook, because this explanation they don't give in your book. You can just note it down. Eugenics is the hygiene for future generations. Then euthenics is hygiene for present generations. The eugenics deals with improvement of human race by genetic engineering. Genetic engineering. You can note it down, this explanations in your textbook, where you can understand better the coming topics. Okay. So next topic is the one multiple alleles. The genetic segregations in Mendelian inheritance reveals that all genes have two alternative forms. The genetic segregation, the genetic separations, that is we call as segregations in Mendelian inheritance, reveal means find out that all genes have two alternative forms, that is dominant and recessive alleles. In Mendelian inheritance, they find out that all genes they have alternate form, that is the one is the dominant, another one is the recessive alleles. Example, tall versus dwarf. So a tall denote as a capital T, dwarf denote as a small t. So that's what they say here. A gene have two alternate forms, that is one is domain, that's a dominant, and another one is a recessive one, recessive alleles. The former is the normal alleles or wild alleles, or later the mutant alleles. Earlier the normal alleles were there. Earlier, former means here earlier, the normal alleles were there, then or wild alleles were there, and later the mutant alleles are there. Okay, the normal alleles means capital T, small t, that is our normal alleles. And wild alleles are just wild bigger alleles, dominant recessive characters. And mutant alleles means some kind of alleles, any one alphabets may be missed out, that is because of mutant alleles. A gene can mutate several times, producing several alternate forms. A gene can mutate. What do you mean by mutate? One gene may be extras, or one gene may be lost. That is because of mutate. Several times to produce several alternate forms. When three or more alleles of a gene that control a particular trait occupy the same locus on the homologous chromosomes of organisms, they are called multiple alleles, and their inheritance is called multiple allelisms. They'll ask you 2 marks, just mark it out. What is multiple alleles? Just start from when 3 or more alleles of a gene that controls a particular traits. So in a gene, 3 or more alleles will be there. That is because of multiple alleles. So their inheritance they call as a multiple allelism. Their inheritance they call as a multiple allelisms. The next topic is the one human blood groups. Human blood groups. Multiple allelisms occurs in humans, particularly in the inheritance of different types of blood groups. Inheritance of different types of blood groups. What is inheritance? Transferring to the next generations, or just having next generations. Through that one, they have different types of blood groups. The blood groups inheritance human can be understood by learning about antigen and antibodies. The blood group of the human can understood by learning of antigen and antibodies. The composition of blood, different types of blood groups, that's ABO. The blood antigens and antibodies were discussed in this chapter 7 of class 11th standard. Last year, 11th standard itself, in chapter 7, we have discussed about what is AB blood group, what is antigens and antibodies. We have learned. Okay, we have discussed in the chapter 7, 11th standard. So, next topic is based on this, based on the human blood groups. The subtopic of human blood group is one AB blood types. AB blood types. Here, what is AB blood types? We are going to see multiple alleles, inheritance of ABO blood groups. What is multiple alleles in the single genes? 3 or more alleles will be formed. That is because of multiple alleles. The blood differs chemically from person to person. Okay, blood always just they have different in chemically from person to person. When two different incompatible blood types are mixed, what incompatible? What is incompatible means? Not matching one. When the human blood which are not in the same blood group, if we mix that one, that is for example, a person who is having blood group AB positive, okay, he can get the AB blood group, AB persons, person who is having AB blood group, he can get blood from all other blood groups. For example, another example, for example, blood group A persons cannot mix this A blood group persons with the AB blood group. Okay, here that is we call as a incompatible. If a person is a blood group A, means the same type of blood group can be transfused. If it is not transfused, means that is because of mismatch. That is what we call as a incompatibility. Blood types are mixed. What will happen? Agglutinations will happen. Agglutinations of erythrocytes. What do you mean by agglutination? Agglutination. Agglutination, which means clumping together. Agglutination means clumping together or just clotting one. That is we call as agglutinations. When two different types of blood groups are mixed together, they undergo further agglutinations of erythrocytes. What do you mean by erythrocytes? RBC. The RBC undergo further clotting. That is the say as agglutinations. The basis of these chemicals differences is due to the presence of antigens on the membranes of RBC and epithelial cells. There is a scientist name called Carol Landsteiner. Scientist name called as Carol Landsteiner discovered two kinds of antigens called antigen A and antigen B on the surface of the RBC of the human blood. There is a scientist name called Carl Landsteiner. He has discovered two kinds of antigen A and B on the surface of the RBC human blood. Based on the presence or absence of these antigens, three kinds of blood groups are formed: type A, type B, and type O. So based on presence of antigen A and B, there are three types of blood groups are formed. That is we call as a type A, type B, and type O. Here they say type O. Let's say blood group A, blood group B. Type A means blood group A. Type B means blood group B. Type O means blood group O. So O, blood group O is called as a universal donor. So this O blood group can donate to any other blood group. That is the universal donor were recognized. The fourth and rarest blood group is the one AB. blood group. The AB blood group is called as a universal recipient. What is universal recipient? The person who is having AB blood group, he can receive the blood from all other different types of blood group A, B, O, and also AB. That is why it's called AB blood group is called as a universal recipient. They'll ask you one word, important. So O blood group is called as a universal donor. This universal recipient was discovered in 1902 by Landsteiner students Van de Castel and Sturley. These two scientists only, the students, the Landsteiner students Van de Castel and Sturley, they identify this AB blood group, the rarest blood group. It's called as a universal recipients. With this, just we'll wind up our class. Just go through whatever we have learned today. If you have any doubt, you can contact me through WhatsApp. Okay, bye. Take care.